Hello.
Right.
Howdy.
Well, any questions about last class period stuff? Lateral gene transfer, bacteria versus archaea.
Remind me, lateral gene transfer is when one bacteria passes it to another bacteria via that tube.
Not just that.
Okay.
That's a special case. So am I sharing my screen? No. Let me share. So lateral gene transfer, aka horizontal gene transfer, is when two organisms who aren't parent-child pass genes to each other somehow. Doesn't have to be with the conjugation, this pilus. Doesn't have to be from bacteria to bacteria either. Okay. So, for example, human genomes, a large portion of your genome is actually viral DNA. How did it get there?
Virus got hungry. Just kind of slurped it up.
Nope.
Is it through transduction?
Transduction, yep. And so it doesn't have to be bacteria to bacteria. A virus can transmit DNA into your genome also. Okay, so that's lateral gene transfer. A lot of the root crops that we eat, like sweet potatoes, they're over 50% bacterial DNA. It's still a sweet potato, but they live in the soil around all these agrobacteria, and horizontal/lateral gene transfer can happen between a lot of different species. Okay. So hopefully that kind of clears that up. It doesn't have to be conjugation. It doesn't have to be bacteria. It's most common in bacteria, for sure, because there's just more of them. What is a plasmid?
A plasmid is a piece of DNA that isn't necessarily attached to the actual the chromosome, right?
Yeah, you can almost think of it like a tiny little chromosome. And so this is just a schematic of a plasmid. It contains like just a few genes. Okay, so it's just some stuff. And this is another reason why horizontal gene transfer is so common in bacteria, is that they've got circular chromosomes, and so these little circular plasmids can be read by them really easily. And so these plasmids can be floating around, they can be sent via the pilus, the conjugation tube, they can be carried in and out by viruses or whatever, but plasmids give like a new set of instructions to the bacteria that's not in its regular chromosome. So it's like having bonus abilities, superpowers, if you will. So that's what a plasmid is. It's very common that in this transformation type of lateral gene transfer, it's actually not little short. I don't like how they drew this. It's not like a little double-stranded linear piece of DNA. It's a plasmid that gets moved around between bacteria. This is where we left off last time, talking about chloroplasts and mitochondria and how they actually are just like enslaved bacteria, essentially. What were we saying about this phylogeny presented here? Maybe a bit hard to read here, but all the stuff in red here are plastids, and the stuff in red over here are mitochondria.
So what's the difference between a plastid and a plasmid?
Plasmid is that circular piece of DNA that bacteria can move around.
Right.
A plastid is mitochondria and chloroplasts.
Oh, okay. Is that so? If it's a plastid, does that mean it became an endosymbiont?
Yeah.
Okay. Got it. Thank you.
Mhm. All right, so I'm looking at this, and we've got nostoc, anabina. Those are bacteria. They're cyanobacteria. Okay. They're closely related to the plastids found in red algae, diatoms, green algae, and plants. Okay. So this, like all phylogenies, is a hypothesis, and like any good hypothesis, it's going to make testable predictions. All right, so what is it hypothesizing here? What's a prediction that it might be making? Anyone?
Isn't it just predicting it's predicting the ancestry and predicting common ancestors of known organisms.
Sure, but I mean it does more than that, right? Like if we saw our phylogeny of like birds and dinosaurs and said, okay, well if that's actually their closer relative, then they should have some stuff in common, right? So we made predictions by that. This is a hypothesis based on phylogeny of genomics, right? So we look at the genes and build a tree, and honestly, this is a bit surprising, right? You take a plant, bust it open, and sequence a chromosome in there, and its closest relative is cyanobacteria. That's kind of weird. So what is this phylogeny actually saying? Big picture about plants. All right, and we take your cell, bust it open, grab a mitochondria, sequence its genome, and turns out that your closest relative is the SAR11 group of bacteria, not chimpanzees. So think like a biologist. What does this make you think of?
It's just like us with the mitochondria, that the chloroplasts became an endosymbiote. Yeah.
And what is an endosymbiont, according to this?
A cyanobacteria.
Or an alpha proteobacteria? Yeah. Okay, so this is saying, if we're to believe it, that your mitochondria are actually alpha proteobacteria, and that a plant's chloroplasts is actually what? Cyanobacteria. Okay, so that's weird. We've got endosymbiosis. So here's the theory of endosymbiosis. Got full screen for you. We're saying that we've got this eukaryotic cell, and it's swallowed an aerobic bacterium that became mitochondria. Okay, if that's the case, should there be other things in common between mitochondria and alpha proteobacteria aside from just the genome? If so, what? What do you know about bacteria? Somebody's talking, I can't hear you, just some muffled sounds. Trying to get you to think like a biologist here and move outside of the simple recall and synthesize some information.
Well, if it is an endosymbiont of a bacteria, then it's going to have all the characteristics of a bacteria, like it's not going to have a nucleus, but
Okay. No nucleus.
Yep, it's going to have the peptidoglycan walls that can be tested
Peptidoglycan.
positive. Circular DNA.
Yep. Okay, so these are the sorts of things that this phylogeny is literally predicting, saying that there should be some commonalities between the plastids here and cyanobacteria. And so one of those is what kind of linkage is in the cellular membrane. Go back. Ester. Okay, so we should be able to look at the membranes of cyanobacteria and see ester linkages. We should also see the same thing in mitochondria. Okay. How does this happen, this process? A cell just swallowing a bacterium and then it's staying around and becoming part of the cell. Well, yeah. Nope, I thought somebody had a comment there.
If it stays part of the cell, wouldn't that normally indicate a mutualistically like beneficial relationship? So like
Definitely.
that's why it stayed like that, because it benefited both organisms.
Yeah, definitely.
If it didn't benefit both of them, it probably wouldn't stay like that, is my thought.
That's a very, very good thinking. Okay, so this should be mutualistic. What is each partner getting out of this relationship?
Usually it's what the other one produces.
Okay, so the mitochondria are producing what?
They synthesize ATP and the cell uses ATP.
Yeah, so they're energy factories. And what do they get from the cell?
The food or nutrients necessary to do it.
The nutrients, yeah, they're getting that carbon, nitrogen, all that good stuff, and they're getting a protected, enclosed environment. They're not out there in the harsh world. They're inside of this nice cell that's feeding them stuff. They're making more energy than they can use. That's getting released, and so we've got free ATP to do work. Okay, so this is a mutualistic relationship. It didn't start out that way intentionally, right? If you've got like a little amoeboid cell, they can actually chomp around these things. They make a living by eating bacteria, okay? And an amoeba eats bacteria, encloses it in a food vacuole, goes into the cell, and then it starts being digested. Okay. Well, this can happen if the amoeba cell ate the bacteria and then got indigestion, just wasn't able to digest it. That bacterium could still be living and producing as long as it's not being killed actively by the cell. So it's sending out energy, they start benefiting. And so now the cells that get indigestion when they eat alpha proteobacteria suddenly are better off. Okay, so that's the environment selecting for this because it's mutualistic. So that's all it takes to get this is indigestion and some time. Okay. Over time, we've actually been stuck here with bacteria in our cells for so long that they can no longer survive outside. And so if you take one of these strains of alpha proteobacteria that is now an endosymbiont, take it out of the cell, it's not going to make it. Okay? Because some of its genes have actually been transferred over into the nuclear genome of the cell. It doesn't have to have them in here if the products that it needed to make, those proteins, are being made by the cell. Okay, so lateral gene transfer can even happen inside of the organs of the cell between the mitochondrion and the host nucleus. And so we've seen that. We find bacterial genes inside the nuclear genome. Okay. And they're the same ones that are missing from the mitochondria that it needs in order to survive outside of the cell. Right, so we're starting to get a complete picture, a lot of lines of evidence pointing to the same thing, is that mitochondria used to be free-living bacteria, and they've been hosted by cells for a long time now. So somebody mentioned that this was mutualistic. That is a type of symbiosis, okay? And we briefly talked about this before, but symbiosis is a spectrum of cooperation. It can range from parasites to mutualists. The mitochondria, or any of the plastids we're talking about, they are mutualistic. Both organisms are benefiting, and in fact, they're acting as one organism at this point. They've been there so long. Okay. Another type of cooperation is metabolic cooperation. So this gets back to our, the biochemical processes that we started talking about. Okay. Pictured here and artificially colored is scanning electron micrograph. Can anyone guess what this is of?
Skin.
Oh, you're close with skin. It's poop. That's human poop. All right. And the different bacterial strains are all just colored differently so that we can see them. They're a consortium of different bacteria doing different things. The waste product of one biochemical reaction might be the starting point for another one. And no single bacterium can do the whole range of transformations that we may want them to, or it may want to. But if you get a bunch together, it's like kind of tag teaming. Right? So this image here, down at the bottom, is showing how you can go from one compound all the way to another, and it doesn't have to be in the same organism. So we can take ethanol, and that can be metabolized by anaerobes into CO2, which can be used by methanogens to create methane. All right, and so these biochemical pathways that we think about, like how do you turn grass into fuel for your car? Well, it takes more than one species. So that's metabolic cooperation. We see this in nature all the time. Okay. One of the best examples, it's shown in your book, are these stromatolites. And they're famous because in the very early Earth, they were the dominant life form. There's just a few places left on Earth right now where they exist. Okay. But stromatolites are these layers of different bacterial species. Okay, so there's an anaerobic layer that can't tolerate much oxygen, and it's sealed in by this green layer. Okay, and so it's just kind of growing, and they're doing each other a favor. All right, so all these prokaryotes, if you stick them near each other, they can use each other's metabolic products. If you grow them all together, it's the same thing, or you can layer them and get really complex consortia. All right, questions about endosymbiosis or metabolic cooperation?
I do have sort of a question. So I've been
Okay.
reading the Your Choice book, I Contain Multitudes.
Yeah.
And I got to the part where he was talking about how lateral gene transfer, or HGT, can actually occur inside your own gut, because he really hits hard on the gut over and over again.
Yeah.
And he said from that the idea of implanting someone else's bacteria into your own can actually cure many diseases. Is that right?
Totally. I mean, you're talking about pretty like bleeding edge science right now in medicine. The idea of transplanting microbiomes from one person to another. And of course, we started out doing kind of like the mouse model because it's, you know, nicer to test mice than humans. But they were able to take mice that were obese, and just by giving them a transplantation of their microbiome, not changing their diet or exercise at all, they became fit and healthy again. Okay. As recently as 2 years ago, I think, a big study of humans came out. I believe it was from Norway, where they actually tracked this, looking at heart disease, obesity, and gut microbiomes. And they actually are at the point now where we're starting to do these transplantations in humans, and they're showing great promise. One of the major areas right now is with Crohn's disease. Right? Nobody really understands what causes Crohn's disease. It's thought to be genetic, but you can transplant the gut microbiome from a healthy person into somebody with Crohn's disease, and it's looking like it's fixing people, curing them of Crohn's disease. The problem is that it's still kind of in its infancy, and we're doing poop transplants. And as horrifying as this is, if you had Crohn's disease, it might be worth it. They take somebody from your household, so similar diet, similar lifestyle, and they collect their poo and they freeze dry it and try to make it safe, but put it in capsule form, and then the sick person takes poop pills. Pretty great, right? So the idea being is that,
this
yeah,
oral form or suppository?
either.
Okay.
Yeah,
Awesome.
they're doing both now. Medical schools are just now starting to like teach this. So if you go to a doctor that's been out of school for 10 years, they're probably not going to be on board with the poop pills because, you know, they get busy seeing their patients and they don't have time to keep up with the literature and all that stuff. So this is pretty new. It's still definitely kind of in the experimental phase, but it's showing great promise, and it's kind of horrifying. But that's, you know, that's the power of prokaryotes. These bacteria, is they shape more about our body, our human phenotype than we give them credit for. And it's because of this metabolic cooperation. So if you get the right species living together in a consortium with the right ratios of them, that has a profound impact on your health because they're able to take products that you're eating and break them down properly, right? The majority of digestion that we do is actually bacteria doing it for us living in our guts. All right, any other questions?
Enslaved bacteria is the best bacteria.
As long as it's good, right? I mean, and in the right spot. E. coli is really great for us if it stays where it belongs. If you eat it, you're going to get really sick. We don't want it in our tummy. We want it in our intestinal tract.
I have a quick question.
Mm-hmm.
So is metabolic cooperation a type of symbiosis, or are they two different things?
Yeah, it is a type of symbiosis because it essentially, you know, the organism here on the right is eating the poop of the organism on the left. And so if you're taking ethanol and converting it to CO2, you're getting energy in the process, and CO2 is your byproduct. That's your waste product. And so you don't even need it around anymore. But the methanogen here can take that CO2 and convert it to methane. And in the process, it's getting its energy and doing its thing. And so altogether, this cooperation is a symbiosis, and it's mutually beneficial. So it's a mutualistic thing. One organism removing the waste product of another and using it. So it's getting the food it needs, and it's cleaning up the environment for the other one.
Okay, thank you.
Yeah. Please note that endosymbiosis is also symbiosis. It's mutualistic. Right. Okay. Well, let's just go on a brief tangent then about why we should care about prokaryotes. Pictured here, you may have seen this out hiking at some point. It looks like orange sludge growing in kind of bogs, really wetland areas. That's iron-fixing bacteria. We call it bog iron. It is the ultimate source of all the weaponry that the Vikings used. They smelted this stuff. Okay, so bacteria are taking trace amounts of iron out of the environment, concentrating them. That's something that we still use today, bog iron. We've got bioremediation. Okay. So the Deepwater Horizon oil spill, I don't know if you guys remember that, decade ago, but very big, horrifying deal, all right? They used genetically engineered bacteria to go in and break up the oil. Okay. We've got bioprospecting. Bioprospecting is awesome. It's one of those like just super cool parts of biology that everyone gets into. So this is going out into nature, looking at these really complex, highly evolved biochemical transformations. Most of them are done by bacteria and fungi, right? And so those are the ones that people tend to study because they do all the coolest stuff. And you're looking for things that are useful or interesting to humans. Prospecting out in biology. Pictured on the left, we've got this bananas title here, cold adapted beta-galactosidase from the Antarctic psychrophile Pseudo-Ultramonas haloplanctus. Okay, that's a pretty big mouthful of a paper title, but it resulted in lactose-free milk, right? We knew how to remove lactose from milk before, right? Lactose is a sugar, so we can break it down, but every process we had involved heating up milk to high temperatures, which made it less shelf stable, all that good stuff. Okay, so some folks went down to Antarctica. They wrote a National Science Foundation grant, and their grant was essentially, we want you to pay us to go to Antarctica to dig up some dirt and sequence DNA from it. That's pretty much their story. And they said, we want $1 million of taxpayer money to go down and sequence genes from Antarctic soil.
And they said, heck yeah.
And they said, heck yeah, because these people didn't just frame it like that, obviously. They said, hey, we are bioprospecting. Okay. And
We're looking
one of the
for
things
some
they
funky
found,
letters.
Yeah. One of the things they found was this cold adapted beta-galactosidase. Well, that's interesting. Okay, so they found it there just like, doing these metagenomic surveys of Antarctic soil. And it turns out that there's some bacteria down there that also can digest lactose, but it's adapted to live in Antarctica. And so this particular version of that protein works at cold temperatures. We could not have invented that given 100 years in the lab, right? Bacteria down there have had millions and millions of years to tweak it through natural selection and mutations, piece at a time. Bingo. So we see that sequence. We can put that sequence into E. coli, and it's going to make that same protein, and now we can use that protein. This is $4 million a year in taxpayer money returned to the United States just from this particular study. Okay, and so that was in 2001. This got kicked off about a year or two later, like actually using this stuff. And so, you as a taxpayer may have paid a hundredth of a penny towards this, but you've got it back, 100-fold at that point. You've earned a penny as a taxpayer because of this kind of basic research in bioprospecting. Another famous one, this one, PCR, won the Nobel Prize. Okay. Polymerase chain reaction, you've probably heard of that, hopefully. It's a way that we can amplify DNA in the lab. And DNA polymerase is the stuff that copies DNA. Well, that's great, but we wanted to copy DNA, melt it apart again, anneal primers, copy it again. So we're making billions of copies of a single piece of DNA. So all DNA sequencing technology up until the last year or two is based on this process. And still a lot of it is. You want to amplify DNA. Forensic science, criminal forensics depends on this. This is how you determine parentage, all that kind of stuff. It's based on this Taq polymerase. Taq stands for Thermus aquatis. That is a bacterium that lives in hot springs. Okay, well that's useful because, we wanted DNA polymerase that can take being at 98 degrees Celsius, almost boiling. No other organism could do that we found. So we went to hot springs and we figured, okay, the bacteria in these hot springs are still amplifying DNA. So maybe their DNA polymerase is going to work at high temperatures. Sure enough, it does. So now we can clone this, right? This little plasmid, stick it into some E. coli bacteria, and now they make this heat-proof Taq polymerase that we can use, right? Also medicines, right? Vast majority of our medicines come from fungi and bacteria. So streptomycin, seen here, antibiotics. But I think one of the most important for the entire life cycle on Earth is just nutrient cycling. Okay. Without prokaryotes, we're all doomed. The carbon and nitrogen cycles break down and everything becomes mineralized. Okay. What I want to briefly talk about is nitrogen fixation. Okay. Most of our atmosphere, when you breathe in, it's mostly nitrogen. Yep, but it's inert. It's N2. Plants can't use that. Right, so what do plants use? They need organic forms, not this mineralized N2 form. All right, so we need to give plants organic nitrogen. That's fertilizer. Okay. So in the 50s, we invented this Haber process, right? And this is a way to turn air into fertilizer. And it's not super efficient. You get 15% of the nitrogen you put in comes back out. It uses a ton of energy. 2% of the world's entire energy budget is used to power the Haber process to make fertilizer out of air. Okay. This is why our human population began skyrocketing. It's because now we can feed people. So this is an important thing, right? We're not wearing out our soil as fast. We can just put this fertilizer into it. Okay, so you heat up a bunch of air at high pressure, really, really hot with an iron catalyst. You throw in your nitrogen from the air, some hydrogen from natural gas, and you can get liquid ammonia out of that. Bingo. So you made fertilizer out of air. But bacteria, they're the only things in nature other than humans that can do this, right? Because they've got this nitrogenase system and these NIFH, the nitrogen fixing proteins. It's a whole lot more efficient. And if you're not fertilizing the area, this is the only source of nitrogen for plants. So you look at the taiga forest that circles the entire northern hemisphere up towards the Arctic. That's a lot of trees. Nobody's out there putting tomato feed on them, right? Ammonium nitrate, none of that. All of their nitrogen comes from bacteria. Okay. The problem with this is that it's slow and it's not enough to feed 9 billion people. Okay.
So
But-
what? Question
yeah.
about that. Is there like a limiting population size? So like if you were just to stick quadrillions of these bacteria into a silo and feed them the amount necessary, it's slow, but I would think unless there's a population limit, it could produce enough. It would just take a little bit to get there to make that constant production.
Yeah, I mean, there's some trouble with this. The NIF-H protein needs to be in anaerobic conditions. So you saw this with your Nostoc, the cyanobacteria. It's got that little cell, the heterocyst. That's anaerobic. That's where it's fixing nitrogen. And so it's a bit more complicated. And at this point, it's just easier and lazier to build this big polluting factory that makes fertilizer than to kind of go the biotechnology route. Okay. I mean, there are other ways of doing this, though. You can do crop rotation, plant soybeans every other year. They're nitrogen fixing. They've got these little nodules where these bacteria live, produce fix nitrogen, and then you can till them under afterwards. So there are better ways to have more sustainable agriculture, for sure. I don't know that any of them is going to be good enough to feed 9 billion people on planet Earth, though. We do have limited arable land space.
Is it fixing nitrogen mostly from the air, or is it recycling some of it from the soil as well?
It's from the air. Yeah.
Okay, so you're not like running out if it's not perfectly efficient.
No, and it's going to go back into the air after you use it. And so you put nitrogen on the ground, bacteria in there are actually turning that back into N2 through this denitrification process. So it's part of that cycle. Now, what about the carbon cycle? We haven't talked about that one yet.
I'm guessing that one would be mostly in the ground.
All right, so
recycling.
Yes and no. Where do you get your carbon?
Food.
Food. Whether you eat cows or soybeans, it all comes from plants because cows are eating plants. That's where they're getting their carbon. All right, so you're getting your carbon, since you're a heterotroph, you're getting it from autotrophs, like plants, cyanobacteria, all that stuff. Okay, so a plant takes air and turns it into sugar. Right? Nitrogen-fixing bacteria are taking air and turning it into ammonia. Right? Both of these are necessary for plant life to flourish, which means it's necessary for you to eat and for there to be any animals on planet Earth. All right, so the carbon cycle goes through plants, then you eat the plants, and then you use that carbon to do stuff. You lose energy as heat, and you exhale CO2 as your waste product back into the atmosphere where plants can eat it again. And so there's the cycle right there. But it's kind of tricky. It's maybe a little counterintuitive, but plants aren't actually responsible for the carbon cycle. What is?
The sun?
What organism?
Oh, it's chloroplast.
The chloroplast, and that's bacteria. And so all carbon cycling on Earth, all photosynthesis is cyanobacterial. Plants never invented a way to do photosynthesis. They just stole it, borrowed it, I guess, from bacteria. Okay. And so bacteria are hugely important at really large scale stuff, things that we actually care about as humans. All right, one last slide. This is just a few years ago. This is to demonstrate how little we still know about bacteria. We got recovery of nearly 8,000 metagenome assembled genomes substantially expands the tree of life. Okay. Well, people went into some extreme environments, some soil, Antarctica, near volcanoes, all this stuff, and they took these samples and they just sequenced every piece of DNA they could find in there, stick them back together computationally, and they found 8,000 new genomes that don't fit anywhere else in the tree of life. This is crazy, right? And they were finding stuff that's so different. It's like the equivalent of finding a new type of animal, right? That's got 50 million legs and breathes fire, right? We just discovered dragons. That would be groundbreaking, earth-shattering news. We found 8,000 new such things, and it gets relegated to a scientific paper. I mean, scientists got excited, right? So we're still finding new phyla of bacteria, brand new branches on the tree of life. And so what can they do? What types of proteins can they make? What metabolic pathways are they capable of utilizing, right? So we know of fungi that can feed on gamma radiation. That's pretty cool, right? So they can be used to clean up nuclear radiation sites. It grows as a black crusty fungus, feeding on gamma rays, okay, absorbing them. So that's pretty interesting. What kind of stuff out there in bacteria land exists that we don't yet know about? And so it's a really exciting field of research, microbiology and, you know, genomics, metagenomics, all this stuff, because we're finding life-changing metabolic pathways out there, right? You could be the person to figure out how to grow insulin in bacteria. It's something that cool that saves thousands of lives. It doesn't have to be, you know, making tastier lactose-free milk, although that's economically very beneficial. Who cares? It's just milk. Milk's kind of gross. So there's stuff out there to find. All right. Now, we will not have class on Friday because that's exam day. And I want to give you that time in case your schedule's busy that you could take it during our normal class. Don't forget to take the exam and don't bother showing up here for class. See, I'm just now seeing the chat. Oh, okay, neat. Yeah. Any questions about bacteria archaea stuff that we've covered?
I had a question about with the mitochondria. Is there anything that they're doing to actively not get digested, whether it's like a mucus or signal proteins on the membrane, or is it a different membrane, or is it just like it didn't get digested?
I think that information is kind of lost to history at this point, because we're talking about over 1 billion years ago when this happened. So I think the simplest explanation is just that it didn't get digested. Now, it still retains its bacterial membrane, right? And so it's actually got 2 plasma membranes now. It's got a bacterial one and then a eukaryotic one on top of that, because it used to be a food vacuole. And so we know that, like, they got engulfed by the same process that proto-amoeba were eating, but I don't know that we have a good answer as to what specifically happened after that, or how it did.
Perhaps add that to the newsletter.
Nothing currently remaining, though.
No.
The employee
Yeah,
newsletter.
they're part of the cell now. They've got a eukaryotic membrane around them, so they're just like any other organelle. We do see big chunks of their genomes have been transferred into the nuclear genome, the eukaryotic chromosomes. The kinds of things that we see tend to be similar for all plastids. They've kind of outsourced a lot of their protein making machinery, that kind of stuff. So they've really been stripped down into just the respiration part for mitochondria and a few other things. And for chloroplasts, they've been stripped down to just the photosynthesis part of cyanobacteria. All the genes are still there from the cyanobacteria. They just aren't in the chloroplast anymore necessarily. A lot of them have been moved to the nuclear. All right, any questions about other material for the exam?
Yeah, I did have one question about open and closed thermodynamic systems.
Yeah.
So, when I looked it up just for review, and it said a closed system is something, or an open system is energy and matter can enter into it, and a closed system is only energy can enter and leave it. Is that right?
That sounds okay. When I'm thinking thermodynamics, I'm thinking energy, not matter, though.
Right.
Yeah, and so the definition I'm working with and the one that biologists I know are working with, it's all about the energy.
So in a closed system, can energy enter?
No.
Okay.
Yeah. And so the reason that we're considered an open system, life is because energy is getting in.
And when I remember you saying that there's very little examples of an actual closed system, the biggest one being the actual universe itself. Is that right?
Sure, and I'm just, it's my failure of imagination to like be able to think of one. I would go to a physics professor and ask them, and you're going to get better answers than from me.
Awesome. Thank you.
Yeah. All right. I will not see you Friday. Oh, yeah.
Sorry, I just have a question regarding the books. So I chose The Ancestor's Tale, and it's a pretty big one. I was curious if we don't have it completely finished by midterm for the Your Choice points, then do we just continue working on it and then get it in after, or?
You owe me 50 points by midterm, so maybe come up with a backup.
Okay.
You can do some of the shorter projects to get your 50 if you need. Or just read faster. That's not always possible. You got other things to do. But if you give yourself like 3 or 4 hours, man, that book is fascinating. You'll sit there and really enjoy it, I think. If you don't enjoy that book, it means that you probably shouldn't be a biology major. I mean, because that means you don't find biology interesting.
Yeah, so far it's definitely been interesting. Yeah, I was going to listen to it, but then it seemed like it was leaving stuff out when I was listening.
It is. Yeah, you can't. That's the one book I know of that there is no unabridged audiobook, so it has to be read, not an audio.
Yeah, so then I bought the book, and then now it's.
You get the pictures that way too, and they're helpful.
Yeah.
Yeah. Have you gotten to the platypus part yet?
No.
Get ready. Those things are insane.
Platypuses are the coolest.
They're awesome. They've got a sixth sense. They can like detect electrical fields. Crazy. All right. Well, I won't see you Friday. I'll see you Monday. Have a good one. Good luck on the exam.
Have a wholesome day, everybody.
Yeah. Nice and wholesome.